The following tutorial will outline a new feature in the upcoming release of the TOXBANK data warehouse. It's called the dashboard, which will allow you to look at multiple investigations or experiments that have been performed in a single view. So once you get to the TOXBANK data warehouse site, the first step is to log in. Once logged in, you'll see the familiar TOXBANK search interface. And for this example, we're going to do a search where we're just going to put in the term liver. So it's going to search for example, liver cells, investigations that were performed on liver cells. And I'm only interested in investigations. So here, if we do a search, it will return the records matching that query. And we see we've got two examples. One is a public data set, TGGate. This is from the TGGates project, the Japanese consortium. This is a transcriptomics data set. And then here is another data set, second one, and this was a data set generated as part of the SURAT project. So if we want to get an idea of how these two experiments were performed, we go ahead and check the two experiments and then click on the dashboard. And the dashboard will summarize these experiments in terms of the biological samples, the treated chemicals, the dose and time points of the treatment. Any protocols that are associated with the experiment will be, there'll be links to the protocols. The protocol documents, as well as the technologies and endpoints in the experiment. So the next step then is, if one of these experiments or one or more of the experiments looks interesting, to go ahead and download that information. So if I click on the download link, what we can see here is more information, and so the full record can be accessed in the first line. Next is the full ISATAB archive, and ISATAB is the universal data exchange format that we're using to format the data, and so it will contain all information on the culture, the protocols. It'll contain information on the study factors as well as the steps that were performed according to, in this case, the transcriptomics. The raw data is available. For example, here are the raw transcriptomic cell files as well as normalized data. And at the bottom here is a derived data, and this derived data is structured according to a proposed standard for derived data, and this contains all the genes with information, for example, on fold changes. So let's go ahead and download this data. Just going to save that data, and then to illustrate how this information can then be used, for example, to understand the biological context, I'm going to open up another tool, and this is a tool called IncreMap, and it was developed at the University of Tübingen. So let's go ahead and load that this transcriptomics data we just downloaded. Here you can see it's already mapped the genes to the ensemble NCBI genes, UniProt. That's performed automatically. Here you can see, because it's structured in the format, the standard, here are fold changes, low at 8 hours versus control at 8 hours. So here we're going to actually just look at the fold change high at 24 hours versus the control at 24 hours. We're going to use this fold change, and we'll use this is the name to describe it. Fold change at high at 24 hours versus control at 24 hours. So it's loading the data into the IncreMap software. It's mapped the genes to their database records. And so what we can start to do now is to explore the biological significance of these results. Maybe as a first step to do a KEGG pathway enrichment. What KEGG pathways have, for example, fold changes greater than 1.5. So if we use that list of genes, 814 genes, what pathways are enriched based on the results from this experiment. So it's highlighted a number of pathways, such as the TNF signaling pathway, Here is values to reflect the significance of that enrichment. Let's go ahead and look at the pathway. Here you can see where the genes are mapped onto this pathway, the ones which are above 1.5 fold change and below minus 1.5 fold change. But what's very interesting then is to go ahead and to color these. So red will indicate significant overexpression. So here I've clicked on this red node, this red gene, and we can see it's 1.8, the fold change, positive 1.8. Click on one of the blue genes, you can see the fold change, negative 1.8 in this case. And so here we're starting to understand the biological significance of the results from this transcriptomics experiment, mapping the data directly onto pathways. So in summary, this tutorial has described a new feature of the warehouse. After searching records, you can then view the experimental conditions and samples and factors in a table, and then access directly the data to use in other applications to do further analysis, to understand the biological context, and also combine the data to support various safety assessment use cases.